All right, in today's lesson, you should have already watched a video. This was the image that was shown in that video about how we go from high pressured arteries to our basically arterioles to capillaries, and then how do we pump that blood against gravity most of the time back through venules, through veins back to the heart. We're going to focus on specifically looking at what is the structure when we look closer at an artery here. So first off, arteries are very strong and they're elastic. They are carrying the most pressure of blood. They're carrying very high pressure. And this is because we're carrying lots of oxygen and we're pumping blood from the heart directly to an artery, trying to get it to every place in the body. And we're carrying with that lots and lots of oxygen. So blood cells are packed in there, and we need those to divide and get smaller. We're eventually leaving through one artery, the aorta, but we're dividing and getting smaller and smaller, and to give rise to arterioles and then eventually a capillary. So as we divide, each time an artery divides, it becomes a new artery, a new name, but also that pressure is going to decrease. The highest pressure is going to be in these arteries. The thickest or strongest is going to be the aorta, and as we branch, it's going to become less thick. There are three basic layers when we look at the wall of an artery. We're going to discuss only arteries today. We'll eventually get to capillaries and veins. But we have our endothelium, which is also called the tunica interna, the very inside layer. So when we look here, remember back to when we studied epithelial tissue, the hole running through the center is the lumen. Okay, lining that lumen, we can see these cells here, and they should look like epithelial cells because that's what they are. The innermost layer is composed of simple squamous epithelial cells. Squamous, simple meaning one layer, squamous meaning kind of a squarish shape. Not cuboidal, they're kind of flat, kind of squarish, and these are allowing red blood cells and platelets to pass through without being damaged. It's very smooth. They don't stick as long as there's no cholesterol in there. And remember, epithelial cells line, so they're lining the lumen in which the blood cells are going to pass through. They also are able to release chemicals that prevent clotting if there's a little tear, okay? We don't want a clot to occur in this layer, so they're also able to release chemicals to prevent that clotting. Again, these are simple squamous epithelial cells, simple meaning one layer thick, and they're made up of epithelium, the squamous flat cells that kind of look like smashed eggs. The outer layer then, if we look on the outside of this lumen, outside of these simple squamous cells, if we're looking in this picture down here, is areolar loose connective tissue. We need to connect that tissue to this middle layer, which is, if you look at my cursor here, is a thicker layer of muscle. So we need a connective tissue to connect the epithelial tissue to the muscle, and so we use areolar or loose connective tissue. So both of those layers, the simple squamous epithelial and the loose connective tissue, are all part of this tunica interna or endothelium that lines the inside of an artery. If we move our way out then, our second layer is the tunica media. Media meaning middle, and so we look, and this is very, very, very thick. If we look at my cursor, all this red down here. This is the thickest part, and it's lots of smooth muscle, involuntary muscle. Smooth, it's not striated, and it's going to control the diameter of the vessel. Remember, we talked about that vasoconstriction and dilation, especially when we're talking about blood clotting and blood clotting factors. So we can constrict this muscle or make it tighten up. We talked about actin and myosin, and that's directly from sympathetic nerve impulses. Or we can dilate that and thin it out, and that makes the lumen larger, and that occurs when those impulses are inhibited, and that's going to directly relate to the pressure inside of there. So if we want to increase the blood pressure, we're going to want to constrict the smooth muscle and make the lumen smaller. If we want to decrease the blood pressure, we're going to vasodilate and we're going to increase the lumen. The last layer then is the tunica externa, and if we look down here where my cursor is, we can see this stuff here. We can see it's white, and we can see like these lines running through it. We did a demo first semester with white colored pencils being like the collagen and these fibers in there. This is composed of connective tissue, okay? It's a very thick, dense connective tissue most of the time, and it's going to attach the artery to the surrounding tissue. So whatever tissue it's running through, it's going to connect it to. We are going to stop there. We're just going to cover the artery today. So we covered the tunica interna, the tunica media, and the tunica externa, and we will eventually lead into capillaries and we'll lead into veins further down the road. But you should have watched the video that was linked in Schoology that described the overall flow of blood from an artery to a capillary to a vein and looked at the differences between the three structures, their structure function. And now we just broke down the three structures that are found within an artery. When you finish this video, please go into Schoology and answer the blood vessel questions. I believe there's about 12 or 13, and wait to be called on to get your poster graded.